Hi, welcome to this lecture on environmental adaptation of animals. Now we will continue on the oxygen and carbon dioxide physiology to look on the breathing of mammals and birds. So far we have learned that in the fish they were having gills as well as in the tadpoles of frogs, but then the adult frogs usually have some kind of lung. But the lung was just a sac, and it was even filled with the mouth structures. But then in the lizards, they were filling the lungs with a suction pump, so using the stomach muscles, etc. But then we have the mammalian lungs, and the branching might be the most typical character on the mammalian lung. It's nothing to do with the sac anymore. Or actually there are plenty of sacs. There are 500 million sacs called alveoli. And the branching is happening several fold. So someone has calculated that there are actually 33 different levels of the branching. So there are the three different kind of tubes there before reaching the alveoli. And that can be easily seen if the isolating the human lungs and injecting plastic, and after the plastic is let to dry, then removing all the organic material. There on the left hand side there are only these airways, and on the right hand side they have used colorful plastic to show that where are the arteries and where are the veins, and you can see that the arteries and veins are also branching in the same way as these airways. And because there are 500 million alveoli, so these microscopic sacs, so the total volume of the alveoli is 130 square meters. That's quite a lot. And all of these alveoli are used for gas exchange. So in the previous slide, everything that we saw was just these tubes, not these macroscopic structures. So the macroscopic bronchs and bronchioles, so that's everything that is visible, they are just allowing the convection of alveolar structures, but not used for respiration. And that's why they are called conduction airways, and they are causing the anatomical dead space. So there are tubes filled with air, but the air is not giving any oxygen to the circulation. And this gas exchange happens in the respiratory airways, where the pulmonary walls are composed with single layer of thin, highly flattened epithelia. So that's why the distance from the gas to the circulation is quite much smaller than in the gills of the regular fish. Okay, how we can measure how much there exist these tubes that are not partly on the respiration? Well, we can measure the breathing quite easily with the spirometer. Even with the very simple old-fashioned spirometer, we can easily measure that, when we are not thinking that we are breathing, so when we are resting, there's about 0.5 milliliter of air is going inside our body and out from the body. And that's called tidal volume. But then we can increase the inspiration by filling the lungs maximally. And several fold more oxygen can be inspirated. So that's why we can then measure the inspiration reserve volume. Or we can try to empty the lungs completely to measure the how much air there at least there exists. So to measure the expiratory reserve volume. So we can have vital capacity, meaning that what is the maximum inspiration and expiration? So how much air can be moved on each respiration cycle in and out maximally. and that's 10 times higher than the tidal volume about. But still, there's air in these airways, and that's about 1/3 of the vital capacity. So there's all the time remaining some air. We can't remove it. And that's why in the normal resting breathing, there is much more air in the airways than when we are having the normal in-breathing. So there's about 2 liters of air all the time in our airways. And that's why we can calculate that the total volume of the airways is about 6 liters, of which half a liter is usually changed during our regular breathing. Okay, so that means that this gas is changed only partly. So when we are breathing half a liter of air, it will be mixed with over 2 liters of expiratory reserve volume, or the residual volume. So there's air that is not fresh. And because there's not, so that's why it means that, okay, there's only 12% of the air is fresh. And meaning that, okay, of these 2 liters, most probably there is less oxygen in the air. And that's why in the alveoles, the partial pressure of oxygen is much lower than in the environment. But then, if we are breathing maximally, we can increase the ventilation, and now up to 80% of the air can be fresh air. And that means that, okay, in that case, if we are breathing maximally, we can have much higher partial pressure of oxygen there on the also on the alveoles. But the problem still exists that because it's a dead end and very tiny tubes, the air is not moving. So the gas is occupying the final respiratory areas. It's completely motionless. And when we are inhalating, the air is moving, but because it's branching so drastically, so the total volume while the air is moving is much larger. So that's why the airflow is ceasing. And that's why it's not moving there in the alveoles at all. And so there's a quite long distance in which the oxygen molecules, carbon dioxide molecules, should be going with diffusion. And this causes that now in the alveoles, there's maybe 60% of the partial pressure of oxygen of the environment. And in carbon dioxide, there is 130 times more carbon dioxide. But the partial pressure is 130 times higher than in the environment. So that's why we humans, we mammals, we terrestrial animals, we are used to have quite high partial pressures of carbon dioxide. So what are the mechanisms of filling and emptying the lungs? So in the reptiles, we are using the muscles in the stomach, so abdominal muscles, and then thoracic muscles on the chest for the suction pump, so enlarging the lungs. But in the mammals, we have a true diaphragm, so there's a sheet of muscle and conduction tissue that flattens when it contracts, and that increases the lung volume. So it's there a bit under. In our case, it's there under the lungs. But then, even in human bodies, we're using intercostal muscles. So muscles that are above the ribs and under the ribs that are affecting on the filling and emptying the lungs, and that especially during the exercise. But that's not the general role in the mammals. So in large four-leg mammals, they can't move the ribs so much. So that's why they are more rely on the diaphragm. Okay, we can move these muscles and then we can increase or decrease the breathing, etc. But how it can be regulated? So the partial pressure of oxygen and carbon dioxide in the alveoli is very constant. And that's why there's no idea of having the regulation only in the to measure how much what is the partial pressure of carbon dioxide of oxygen in the air in the alveoli, and it's also difficult to measure from the air. And the respiration control happens mainly by measuring either carbon dioxide or pH with the chemosensor regulation. In the brainstem, there are some nucleus that are affecting it. And they are very sensitive. So when we are having 10% more, 10% higher partial pressure of carbon dioxide, the ventilation is doubled. So we are very sensitive for measuring how much carbon dioxide we have there. And why we are measuring the pH is that, okay, in the previous videos, we were discussing that the carbon dioxide is reacting with water and causing acidification, so it's forming carbonic acid. But the good point is that, okay, now this ventilation is also related on the glycolysis. So when we are not having oxygen available, and we are having this anaerobic catabolism and producing lactate, and then there's more lactate that is lowering the pH, and that causes that, okay, we are increasing the ventilation. But of course, we have oxygen sensors. In our body, we have in chaotic body. In several other mammals, there is also in our aortic bodies oxygen sensors that are quite insensitive. So now the oxygen partial pressure of oxygen must actually drop almost to half before the ventilation is stimulated. So that's why in the normal life we are just measuring carbon dioxide. Okay, how it works in more detail. It works a little bit different in normoxic and hypoxic conditions. So when we are going, for example, in high altitudes, etc. In normoxic, we have a chaotic body that has actually oxygen and also pH sensor. And it regulates the brainstem via a solitary tract nucleus, and one of place where it's affected is called retrotrapezoid nucleus. Very difficult word. Let's call it RTN. And this nucleus is regulating actually 4 different nucleus, including these Bötzlinger and pre-Bötzlinger complex. And some of these nucleus are regulating 4 other nucleus. So that's why you have at least 8 different nucleus that are regulating. And that's why quite often it's called that, okay, you have just the respiration pattern generation. Because you have several different muscles, some of them are skeletal muscles, and they must be regulated. And you are not regulating only that, okay, what is the rate of the breathing? So how fast, how many times per minute you are breathing, but also the inspiration amplitude, etc. So that's why there are several different nucleus. And then when you are regulating the breathing, then, okay, you are producing at the same time more carbon dioxide or affecting on the pH, and that then can be regulated on the RTN nucleus. But also, it can be regulated by some glia cells. So it's more complex than just having one nucleus there in the brainstem. But in the hypoxia, the karyotic body is regulated now with the oxygen sensor. And this affects that now this RNT nucleus is not so dominant. And then we can increase the breathing very drastically and get rid of carbon dioxide and acidification. And that's why we don't need so much on the glia and again, the RNT regulation, etc. So we are using several different nucleus, and in normoxia, we are measuring carbon dioxide or the proton level, so meaning the pH. But then in hypoxia, you are switching off some nucleus to increase the breathing. Okay. How we can boost the breathing then? The overall lane of lining ventilation depends on two things. So what is the volume that we are breathing per one cycle? So the tidal volume, because normally it was half a liter. And then of course the frequency of the breath. And interesting human, we are breathing for every cycle, we are breathing maybe half a liter and maybe 10 or 12 times per minute. So it means, okay, there's gas exchange, not gas exchange, but there's air exchange in the airways for 6 liters per minute. But usually we can't maximize these both. So if we are increasing, for example, the tidal volume, then usually the breathing rate will decrease, because it will take more time for these breathing. And also, when we are breathing very frequently, there is not so huge amount of air going in and out. But then, if we are thinking on the alveoles, we must now think that, okay, how much air can go there in the alveolus? That also affects on the dead volume on these airways, but this dead volume is quite constant because these tubes are quite constant size. So that means that if we are increasing the tidal volume, so in breathing more deeply, then the tidal volume is increasing and more air is reaching the alveolus. And that's why in the resting breathing in humans, we can take maybe 20% of the oxygen from the environment to circulation. But when we are breathing maximally or breathing very deeply, then 30% of the oxygen can be taken. But that's still quite ineffective. So we can't get it all. What about then other kind of mammals than humans? Well, we all know that the small animals are breathing in fast frequency. Over here we have a picture of different kind of mammals, and from the left to right there is an increasing body size. And then from bottom to up there is a increasing breathing rate. As you can see that these rodents in red that are weighing maybe less than 100 grams, they have breathing rate way over 100 times per minute. But then, for example, these larger animals like elephants, they are breathing only few times per minute. And there are some variation. So there's one rodent having breathing rate or less than 60, although it's 100 grams. But actually that's one of these hibernating animals, so it's not very typical in this kind of comparison. And there are some variations also between carnivores and etc. But why these small animals are breathing so fast? It's because the lung volume is constant. It's 6% of the body weight, and the tidal volume in the rest is 0.6% of the body weight. So that's why all the animals are getting equal amount of fresh air to compare their body weight during each breath. But we have learned that these small animals have higher weight-specific metabolic rate, so they have higher oxygen consumption. So that's why they must increase the breathing frequency quite drastically. Okay. What about then the birds? Aren't we just completely the same? No. The lungs of the birds are completely different. We have high body temperature, we have high oxygen consumption. The only thing that is common is that we have trachea that is divided on two primary bronchs. In the birds, they have two sacs. One sac is on posterior, near the tail of the bird, and then one is anterior, more near the mouth, and they fill these sacs, and when they are filling or emptying it, then the air is flowing beside air capillaries in one direction, and so there's moving air in tubes. So the mammalian lung, it actually can be thought as a sac. There's millions of microscopic sacs that are having non-moving air. But in the birds and the crocodiles, they have a moving air in the tubes, and then they have the tubes for these gas exchange from the air to circulation. And these sacs have several different names, but quite often it's just called that, okay, one is anterior sac and one is posterior sac. Then there is a primary bronchus, and then it follows through the lung as a mesobronchus, and beside that there are parabronchus. And on the anterior side and posterior side, there are some names for these tubes also, as well as there are some additional neopulmonary parabronchus that we are going to discuss later. So several different kind of bronchs, but thank God we don't have to worry on the names so much on this course. During the inhalation, the air is moving to this posterior sac and filling it, as well as the air is moving through these parabronchi tubes to fill the anterior sac. So during the inhalation, the air is moving in these tubes, and some of that can be used for this gas exchange in these tubes. And then during the exhalation, these posterior sacs are compressed and the air is pushed again through these tubes, as well as they are pushed from these anterior sacs and out from the animal. So all the air that is flowing during the inhalation is pushed practically through these parabronchi tubes and then participating on the gas exchange. But gas exchange happens during the inhalation and exhalation, whereas in humans the gas exchange happens all the time, but very slowly because the air is not moving. So there is an opening lead to air capillaries in these parabronchi, and they have these very small capillaries there are capillaries of air, there are capillaries of blood, and the total surface is enormous, 250 square millimeter per cubic millimeter. So during this parabranchi, the air is moving on one direction, and then around it there are these capillaries where the gas exchange happens. And then there is this blood flow that is not going on the same direction, not on the opposite direction, but it's going from left to right and taking the oxygen from the air to the circulation. Okay, the sacs are not lungs. They fill the large areas of thoracic and abdominal body cavities, so if you open the stomach, there are plenty of these sacs. And in typical birds, they have 9 sacs, and they are connected by a paleopulmonary system that is described before. And because it's paleopulmonary, it's been there for ages. And these lungs in the birds, they are not moving. You can have quite small lungs in the bird, and they are not moving. So, and this filling of this sac, that's quite simple. So you don't need so much energy for filling and emptying the sac than when having a lung-like structure that must be stretched, etc. And these posterior sacs, there are in several. Most of the birds, they have these neopulmonary system. So they have not only one type of tubes, they have also another kind of tubes. And these neopulmonary system, these tubes are used for singing. Thank you.